All right, ladies and gents, welcome back. So today we're going to finish up our discussion about how cells are capable of using sugars to make ATP. So when we talk about cellular respiration, the overall process is aerobic. It requires oxygen because we need it as that final electron acceptor at the end of your electron transport chain. But there are situations where organisms either exist in an environment without oxygen or they simply never evolved to use it. And so when we look at how they are capable of making ATP without oxygen, that's where this idea of fermentation comes in. So regardless of whether we're talking sugars to ATP with cellular respiration or we're talking sugars to ATP with fermentation, glycolysis still occurs, period. Whether we're talking aerobic or anaerobic, everybody on Earth uses the sugars and goes through that glycolysis process. It's post-glycolysis then that branching occurs. If I'm in an organism that is present in oxygen and capable of using it, it will go through cellular respiration because cellular respiration overall is a very much more efficient process. They're making somewhere on the order of approximately 38 ATPs for every glucose that goes into glycolysis at the start. I get 38 ATPs, give or take, at the end of cellular respiration. But if I'm in a situation where either the organism can't use the oxygen or there is no oxygen present and they are capable of the fermentation, this still allows glycolysis to continue, but now I'm only able to make 2 ATPs, so a much more inefficient process. All right. And those 2 ATPs are coming from the glycolysis itself. All right, so the whole point to fermentation is to allow glycolysis to keep occurring if the cells are capable of doing it in order to make the 2 ATP that glycolysis can produce. Now, when we look at fermentation and we look at organisms that are capable of it, there really are 2 different types of fermentation that exist in nature. So if I look at lactic acid fermentation, this occurs in animal cells, or also some bacteria. By the way, you may want to put that down as a side note. Some bacterial cells are also capable of doing lactic acid fermentation. And they're going to go through glycolysis just as normal. So we're going to take that glucose, go through the energy investment and energy payoff phases, get that net 2 ATP out of it, make those 2 NADHs, and produce those 2 pyruvates. It's then what happens next, and that's where the lactic acid potential option comes in. So for cells that do lactic acid fermentation, they're going to take those pyruvates and create lactic acid out of it. Okay. Now, why does fermentation occur at all? Well, remember that in order for glycolysis to keep going, this is where the energy is coming from. So in an anaerobic situation where there is no oxygen, I've got to be able to reproduce this NAD+. All right, because part of that glycolysis process is taking NAD+ and making NADH. Now, if I'm going through cellular respiration, this NADH gets shelled to your electron transport chain. The electron transport chain pulls off the hydrogens, creating the NAD+ back, and it gets shuttled back here to keep that process going. But if I have no oxygen, I can't do my electron transport chain. And so the fermentation takes that step, or replaces that step, I should say. And so they're going to take, in this process of converting pyruvate to lactic acid, take that NADH, recreate the NAD+, so that that can go back to glycolysis and keep getting used. All right, so fermentation really is about recycling that NAD+, because I don't have an electron transport chain here to do it for me. And so fermentation takes its place. Okay. What kind of cells do this? Well, like I said, some bacteria cells will do this. But I also have an example of this in your own body. Your body can take that pyruvate, convert it to lactic acid or lactate, depending on what you come across. And if you've ever done a really hard workout and felt that sort of burn in your muscles post that workout or even during it, it's because your cells temporarily can utilize lactic acid fermentation to compensate for the slow down, the decrease in concentration of oxygen in your muscle cells at the time. All right. Post that workout, your body's going to have to break down that lactic acid so it doesn't continue to destroy your muscle cells, but it will be able to give you some of that energy, at least for short stints.
So not only can lactic acid fermentation happen in animal cells, like we see in your muscles, but it can also occur in some bacteria. For example, those that end up converting milk into yogurt do lactic acid fermentation in that process, which is why you sort of get that bit of a tang or sort of hit at the end of that bite of yogurt. It's because of that acid that they're producing. That acid sort of has that sour sort of note to it at the end.
Option number 2, if we're talking about cells that do fermentation but it's not lactic acid, then the other option is alcoholic. And this is what we're talking about in certain yeast species as well as some other bacterial species. Again, still in an anaerobic environment. We're still going through glycolysis to make those 2 ATPs. We're still making pyruvate, and the alcohol fermentation is still taking that NADH, recycling the NAD plus so that glycolysis keeps working. It's just a question of how we go from pyruvate to an end product in that recycling process that looks a little different. So with lactic acid fermentation, in order to recreate that NAD+, we go from pyruvate to lactic acid. When we talk about alcoholic fermentation, these guys are going to take those pyruvates, recreate the NAD+ from it, but in that process, instead of making lactic acid, they're making alcohol and carbon dioxide. All right, they're making ethanol, to be more specific. We use alcoholic fermentation in a lot of industrial food processes. The yeast, right? This is why we add them to that dough, because what they'll do is they'll go through alcoholic fermentation, produce the CO2, and that CO2 production allows your bread to puff up and be nice and fluffy and soft. They are producing ethanol or alcohol in the process, but when you go to cook the bread in your oven, that alcohol actually just kind of burns off. It's got a relatively low boiling point, so it just burns off in your oven, and you're not going to get tipsy from eating bread. We use some certain bacterial species who do this to make things like yogurt and cheeses. Right? They'll take that milk, they'll culture these bacteria in that milk and allow them to go through fermentation, and it will turn it into cheese or yogurt. Soybeans into soy sauce, cabbage into sauerkraut. It's all based on this fermentation process that they are capable of doing. And it's really just about those species being able to survive in an anaerobic environment by allowing them to still make some ATP from the sugars that are there. But that fermentation is there to recreate that NAD+ so that the glycolysis.
Hi, it's Mr. Anderson, and in this video I'm going to talk about anaerobic cellular respiration, or cellular respiration without oxygen. To understand anaerobic, you must first understand aerobic respiration. And so if these terms don't make sense to you, glycolysis, Krebs cycle, electron transport chain, and if you don't even know what a mitochondria is, you may want to go watch one of my videos on that, and I'll put a video link right up here. But what is anaerobic cellular respiration? That's when we don't have oxygen or we don't have a mitochondria present. And so let's get rid of those. And so what is anaerobic respiration? It's really just glycolysis and then a new process called fermentation. And so let's dig in a little bit deeper. And so this is all the steps of cellular respiration. Remember, we began with glucose. In glycolysis, we break that down into pyruvate. How much energy do we get from that? We get 2 ATP. Now we put in some ATP, but we net a total of 2 ATP. What happens to the pyruvate? It's going to go into the mitochondria. It enters into the Krebs cycle after it's converted to acetyl-CoA. We give off all that carbon as carbon dioxide, and we make another 2 ATP. And so we haven't released that much energy yet. Where'd the energy go? It's stored in NAD and FAD. They're going to transfer their electrons through the electron transport chain. Eventually, those electrons go to oxygen with the formation of water. And we're going to make most of our ATP here. And so we're going to make somewhere between 32 and 34 ATP. And so we net around 38, but there's controversy. It's probably not as much as that. But how could we break this process? Well, we could break this process, number one, if we didn't have any glucose, but we usually have enough food inside our body. But we could break this in 2 ways. We could get rid of the mitochondria, so if there was a toxin that destroyed the mitochondria, for example, or if we just didn't have enough mitochondria present, or if we didn't have oxygen. Remember, oxygen's right here at the end. It's receiving those electrons. It's the final electron acceptor. And if we don't have that, the whole thing kind of backs up. And so we're out of luck. And we would be out of luck if it weren't for anaerobic respiration. If you want to feel what anaerobic respiration feels like, just hold your breath for a while. You're going to run out of oxygen, you can't make ATP, and you're going to get in some serious trouble very, very quickly. And so what is the problem? Why are you feeling that pain? Well, it really boils down to glycolysis. And so in glycolysis, we're taking glucose and we're breaking it down into pyruvate. Remember, we net 2 ATP. Where did that energy go? It's being converted to NAD. A lot of it is converted to NAD. And so NAD is going to be reduced. Remember, it's going to pick up electrons. But pretty soon, all of that NAD is full. There's no electrons that can be donated to it because it's now all at NADH, or reduced NAD+. And so that's where we get stuck. And where are we going to come up against this wall if we don't have oxygen or if we don't have mitochondria? And so what is our solution to that? Well, through evolution, we've come up with 2 solutions to this. We have lactic acid fermentation and we have alcoholic fermentation. So first you have to do glycolysis, but after that, in animals and bacteria, they do what's called lactic acid fermentation. And so in a sloth, they don't move that fast, but maybe in you when you're sprinting, or in bacteria when they're making yogurt, they can do another process after glycolysis, and what that does is allows us to keep doing glycolysis over and over again. And in alcoholic fermentation, they do that by actually converting it to ethyl alcohol. So let's go through those specifically. Again, here's where we're stuck. We've gone through glucose or glycolysis, we've made pyruvate, but now we have all of this NADH, and there's no way that we can keep going through glycolysis because all of it's filled. And so in lactic acid fermentation, what happens is this pyruvate is converted further into lactate. And sometimes you've maybe heard of that called lactic acid. What happens with the formation of lactic acid? Well, we're not making any ATP, but those electrons can now be converted from NADH, and it can be transferred to lactate. What does that do? It frees up this NAD plus to go back and pick up more electrons again. And so what we can do is through this process, we can go through glycolysis over and over and over and over again. And so we can make ATP every time we do that. Now, we're not going to get all that ATP that we would if we went all the way through Krebs cycle electron transport chain, but we can still make quite a bit of energy. Now, this is a picture over here of my son. He is a cross-country skier. And so in this picture right here, he's on a treadmill, he's skiing. And this is a test to calculate VO2 max, to figure out how efficient you are at using oxygen. But it also is going to measure your lactate threshold. It's going to measure the amount of how much exercise you have to do before lactic acid builds up in your muscles in an appreciable amount. And so if you are exercising really, really quickly, you get a certain amount of energy through cellular respiration. But if you go faster and faster and faster, eventually your body will also add on top of that this lactate acid fermentation. And if you've ever run, for example, a 400-meter dash or sprinted, that pain you feel in your muscles is a buildup of this lactate in your muscles. And so eventually that's not even enough. And you're eventually going to just have to stop running or stop competing because it's too painful. And that's that buildup inside your muscles. And so what happens is after you're done, then you have to go through and breathe a lot and then use oxygen and cellular respiration to break down that lactate. But it does give us kind of like a turbo boost to go on top of that regular cellular respiration. Bacteria do the same thing. If you were to put them in milk, for example, lactobacillus bacteria will go through lactic acid fermentation, and that acid breaks down the proteins in the milk and makes yogurt. And so that's one way that we can survive when we don't have oxygen, lactic acid fermentation. Remember, it still includes glycolysis, but it's followed by this lactic acid fermentation, so we can go through that process again. Now, we also see the same thing in alcoholic fermentation. And so where would we see that? That's going to be in things like yeast. And so what are they doing? They're breaking down glucose into pyruvate, but again, they're stuck. And so, for example, if we put a little bit of yeast and some grain and sugar in this bottle, they're going to start to do cellular respiration, just like we do. But eventually, they're going to run out of oxygen. Okay, no oxygen can get in this container, only gas can get out. And so eventually, they're stuck. And they would be stuck if they couldn't do fermentation. What are they going to do? They're going to convert that pyruvate into ethyl alcohol. And that's the alcohol that we'd find in beer and wine. Now, if you look at pyruvate and ethyl alcohol, we're missing a carbon here. And the reason why is that that carbon's going to go towards carbon dioxide. That's why we'd have a buildup of this carbon dioxide in beer or champagne, for example. What is that doing, though? Again, it's the same thing. It's picking up electrons from NADH, and that's producing more of this NAD+, and so we can go through that process of glycolysis over and over and over again. And so if we're looking at yeast inside here, now they'll do alcoholic fermentation, and they'll do that until they have consumed, built up too much of this ethyl alcohol, and then it'll eventually poison them. And so we've known this for a long period of time. And so fermentation has been going on for years and years and years. The Egyptians used to make beer using fermentation, and we do it today as well. And so what do you need? All you do is put a little bit of grain in there, some sugar, water, and some yeast. If you don't give them oxygen, eventually they're going to convert to alcoholic fermentation. And they'll do that until the level of alcohol inside there is going to kill the yeast. They settle to the bottom, and then we have alcohol. And so that's anaerobic respiration. What does it do? It allows us to keep going if we have no oxygen or no mitochondria present. It only lasts for a certain period of time, and then we're out of luck. And I hope that was helpful.
Keeps going.